Okay, next we have Discovery Lab 2, represented by Grace Engel. The title of her talk is Identification of Novel Chemotherapeutics in Drosophila melanogaster.
Hi, I'm Grace. So today I'm going to give you a brief overview on cancer research and why it's so important. And then we'll transition into what we did this semester and how we applied all of that information to the drug discovery lab. So there are many different causes of cancer. You're probably already familiar with a lot of these, like smoking, not wearing sunscreen, or acquiring sexually transmitted diseases like HPV. There's new research that also shows that your diet can impact whether you get cancer or not too. So to put everything in perspective, please raise your hand if you know someone who's been affected by cancer or have been personally affected by cancer. Look around the room. Almost everyone is raising their hand. This is a nationwide epidemic that affects about 1 in 2 Americans during their lifetime. So now they're all scared. What actually causes cancer? Cancer is DNA damage, and it occurs in normal cells, but usually it's corrected. When it's not corrected, and when it occurs in important genes, it leads to uncontrolled cell division. This is what constitutes a tumor. There are 3 main factors that cause cancer. The first is environmental. These are your behavioral and lifestyle decisions, again, like smoking. There are also random causes of cancer. This is the DNA mistakes that happen in your everyday cells, and it accounts for about 2/3 of all cancer. It's just bad luck. The third is genetic. These are inherited factors. They give certain individuals a predisposition to acquiring certain cancer-causing mutations. So it's been around for a long time. It's pretty scary. So why haven't we cured it yet? As it turns out, every single cancer is different. And within one individual, there might be multiple tumors, and within those tumors, multiple populations of mutations that are each cancerous. This is called tumor heterogeneity. And usually cancer cells start out with 1 or 2 mutations, but as they continue to divide, remember they're rapidly dividing, they acquire more mutations, and this leads to an accumulation of genetically distinct cancer-causing populations. Each of these might require a different approach to treatment, and this is why treating cancer with one single chemotherapy is so difficult. Another challenge we face is drug resistance. Cancer cells are extremely smart, and they learn how to counteract the drugs that we use to kill them. They can do this by pumping the drug out, activating enzymes that break down the drug before it can do its job, blocking uptake, or modifying intracellular targets that change the way the drug works. So why can't we just pump your body full of chemotherapies and see what happens? Well, as it turns out, chemotherapy targets dividing cells in your body, and there are a lot of different dividing cells in your body. So this leads to a lot of unpleasant side effects that can be very dangerous for cancer patients. If this group of people right here represents cancer, sorry, we're trying to kill you in this metaphor, the rest of you represent healthy cells. And administering chemotherapy is the equivalent of throwing a grenade into the audience, crossing my fingers and hoping for the best. It is extremely inaccurate and very dangerous for other living cells. This is why cancer treatment is almost as dangerous as having cancer itself. So I've just given you an overview as to why cancer research is so important, but now I'm going to tell you how it applies to what we do. So we'll go over our experimental aims, methods, and student projects. We use this fly, Drosophila melanogaster, as our model organism for tumor. We're actually interested in these guys right here. They're called third instar larvae, and they share about 75% of their genes with humans, and they behave very similar to a tumor. So in our research, we're actually trying to kill them. Remember how we talked about how cancer can have multiple populations? Well, that's what makes it so difficult to cure. So in this lab, we combine radiation treatment with possible chemotherapeutic compounds in order to reduce the chance of reoccurrence. This can also decrease the toxicity because the chemotherapy and the radiation are working together. How do we do this? Well, we start out by separating our third instar larvae from the rest of the population. Again, these are our tumor cells, so we want to isolate only them. Next, we irradiate the larvae with 4,000 rads of radiation. And just to give you an idea of how much that is, you start to experience uncomfortable side effects at 2. These little guys are 3 millimeters in length, and 4,000 rads only kills about 50% of them. So they are very indestructible. Next, we expose them to the possible chemotherapeutic compounds and quantify survival. Most of our data will fall in this region, about 95%, and that means it actually doesn't do that much to the flies. However, we're looking for data that fall outside of the range of 2 standard deviations from the mean. That's this data right here, and that indicates that it kills an abnormally large of larvae. One of our biggest accomplishments this semester was that we tested 813 compounds from the National Cancer Institute. And again, the data in the lower third portion, whenever it comes back, yeah, this right here. These are our hits. This is 2 standard deviations below the mean. And this means, again, that they kill an abnormally large amount of flies, which, again, represents cancer. So we actually discovered 32 different potential chemotherapies, and 3 of these are already known chemotherapies. So this is fantastic because it means that our research is working. Students also completed an independent project this semester. This is my project. It's an organic compound called sulforaphane, and it's found in cruciferous plants like broccoli. Sulforaphane is usually used as adjuvant therapy, meaning that you can pair it with chemotherapies to increase the effect without increasing the dose. Again, decreasing the toxicity and the side effects associated with all of those chemotherapies. Other students studied CBG and CBD. These compounds are derived from cannabis, but they are non-psychoactive. Most importantly, these can improve motor skills, increase immune function, and induce apoptosis. Apoptosis is also known as cell suicide, and if you're trying to eliminate cancer, this is a really important and helpful tactic. Yet another student studied Cuban blue scorpion venom. This compound can modulate inflammation and cell division. These processes are both exploited by cancer cells, so getting them under control can also help stop cancer. So what's next? First, more flies. All of our experiments must be repeated in order to validate our findings. Next, we will determine a dosing series that will tell us how much of each compound is needed in order to make it effective. Hopefully, we can move these compounds into mammalian cell testing in order to determine how they work, and hopefully we graduate some of these compounds to clinical trials in order to reach real-life patients suffering from cancer. None of this research would have been possible without the people you see on the screen. So I'd like to give a special thanks to Dr. Tin Su, our principal investigator, our fabulous professor and coordinator of this event, Dr. Pamela Harvey, our teaching assistants that dedicated evenings and weekends, and funding provided by the Howard Hughes Medical Institute, Biological Sciences Initiative, and Department of Molecular, Cellular, and Developmental Biology at the University of Colorado Boulder. So he asked if there were any known chemotherapies that were deemed ineffective. No.
That's a great question, right? Because it's a model for head and neck cancers, but not all cancers. So there would be, I think there's one poster here that tested 6 different FDA-approved chemotherapies combined with